Good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled Fragment Based Drug Discovery, Hitting Targets Using the Right Chemistry and Expertise Alliances. My name is Sonia Hunt, and it's my pleasure to be your X Talks moderator for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout this presentation by using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located in the control panel, and that's found on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending me a message using that chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank these companies here who developed the content for this presentation. First, I'd like to introduce OncoDesign. OncoDesign Services is a biopharmaceutical company dedicated to precision medicine, founded in 1995. Its mission is the discovery of effective therapies to fight cancer and other diseases without therapeutic solutions. And next, we have Crystals First, which provides the most advanced structural biology platform to unlock chemical matter in drug discovery by using the SmartSoak, which stabilizes protein crystals for X-ray crystallography. And next we have Turbine. They are the experts in biophysics. They have successfully completed over 1,250 tasks in contract research projects with over 350 customers. And finally, the Weizmann Institute of Science, the world-leading multidisciplinary basic research institutions in the natural and exact sciences. Now, it's my pleasure to introduce you to your speakers for today's event. And first, I'd like to introduce you to, there he is, Dr. Thomas Sherbert. He's a managing director at Turbine. Hello there, Thomas. And next, I'd like to introduce you to Dr. Sergey Glinka. He's the founder and CEO of Crystals First. There he is. Thank you so much. And next we have Dr. Nur London. He's the president of the medicinal chemistry section of the Israel Chemical Society. Sorry, everyone, at the Weizmann Institute. And there he is. Thank you so much for joining us. And next, it's my pleasure to introduce you to Dr. Jan Lamont. He's the chemistry team manager at OncoDesign. And there he is. Now, it's my pleasure to pass over the mic and the controls to our first speaker, Jan. So Jan, when you're ready, you may begin. And I'll pass you the controls right now. There you go.
Thank you, Sonia, for this kind introduction, and thank you everyone to join us today for this webinar. Fragment-based drug discovery is a powerful method for developing potent small molecules, starting from fragments, binding weakly to the target. In 2 decades, FBDD became an attractive strategy in drug discovery, and many preclinical and clinical candidates have been developed using this approach. Today, more than 49 molecules are in the clinics, and the analysis from 2019 to 2021 showed that there is a constant number of molecules in phase 1 clinical trial and small increase in phase 2 clinical trials. In 10 years, 6 molecules were approved by the FDA, and they are all used in oncology. First, Vemurafenib was approved in 2011. This compound targeted the BRAF mutant form. First, in 2016, venetoclax was approved to treat leukemia after ribociclib, which targets CDK4, CDK6 for treatment of breast cancer, erdafitinib, which is a kinase inhibitor, FGFR, a kinase inhibitor, and more recently, just last year, sotorasib, has been approved for the treatment of non-small lung cancer, and it is a KRAS G12C mutated form inhibitor. And finally, asciminib, which is used for the treatment of leukemia. So what is a fragment? A fragment is characterized by a low molecular weight, below 250 daltons, and a weak affinity for the target, with a range from millimolar to micromolar, but strong interaction for the binding site, as defined by ligand efficiency. If we compare FBDD and high-throughput screening, high-throughput screening is defined by potency, but fragment-based drug discovery is defined by efficacy. And the efficacy is defined by the ligand efficiency, which is defined by the free energy binding divided by number of heavy atoms of your fragments. The initial value of ligand efficiency, 0.3, 0.4, need to be maintained during the lead optimization stage. Probably all of you are familiar with the rule of 5 as defined by Lipinski in 1997 to define the drug likeness of molecule. And for the fragments, Aztecs define the rule of 3. Fragments need to have a molecular weight less than 300 Dalton, H-bond donor or H-bond acceptor below than 3, and a relatively low lipophilicity with a C-log P below than 3. And when you look at the chemical space corresponding to the fragments versus the chemical space of molecules coming from the HTS, you can see your optimization space is larger to develop your fragments to a drug candidate. Identification and characterization of fragments as a starting point in a drug discovery program is key. And different techniques are used to identify fragments, and my colleagues from ToBind and Crystal First will show you in the next slide how biophysical or crystallographic methods are used to identify best fragment to initiate a drug discovery program. So I will pass the control to my colleague Thomas from ToBind, and just to show you the characterization by biophysical methods.
Thank you, Jan. My pleasure. So, could you pass me the control? Thank you. So, my pleasure is to talk in the beginning about the fragment-based drug discovery process. As you are aware of the fact that the team of OncoDesign, ToBind, and Crystals First cover the complete process of fragment-based drug discovery. It starts with after the target has been validated, we can assist in hit identification fragment screening, followed by validation of identified hits. The validated hits go into fragment growth phases in which structural information is used by medicinal chemists to grow fragments towards compounds and finally to leads. The lead is then optimized, and finally it will turn into the preclinical regulatory area. At ToBind, we can assist in the rather early phases of this process. We have a rather large portfolio of different tools that we can use. First of all, in the very early protein QC, it's a very essential step. Then in the hit identification, meaning fragment screening using biophysical tools. We can then also assist in the hit validation phase with various methods, and finally also support the medicinal chemists in their SAR process. In the protein QC step, we are focusing on factors like colloidal stability, thermal stability, size, molecular weight, and use methods to determine those. Only proteins that fulfill certain criteria, which basically pass the control, will be used in biophysical screening. We could also add some compound solubility and fragment solubility assays in addition. In the fragment screening approach, using biophysical tools, we have different options, and depending on the aim of the project, depending on the biological system the protein target is originated from, but also depending on protein constructs, we can choose from different tools. We can rank according to affinity using, for example, MST, dianthus, or spectral shift. But we could also go for kinetic ranking using the GCI, which is capable of fast on and off rate ranking. We could also go for a yes/no answer, so it's not a quantitative, more qualitative assay in terms of readout, a thermal shift assay, modification free, and we combine that as we need. We have two fragment libraries here at 2Bind, but of course we can also work with external libraries for hit identification. Depending on the primary screening tool that we have used, we can then validate hits using orthogonal methods and add information on affinity kinetics as the situation needs. In the fragment growth phase, in the MedChem support, the evolution of compounds is monitored, and very often the affinity is here the measure that is chosen. Nevertheless, also kinetic information can be used. For affinity, as mentioned, MST spectral shift makes sense, and kinetics are commonly done with GCI. And in addition, also thermodynamic parameters might make a lot of sense to determine. It can be helpful for medicinal chemists. To show you in the last few slides a little case study here. It's a CNS target that we have screened for fragments. A little bit more than 450 fragments were used. 3 control ligands were included in the assay development phase. And due to the project aim, due to the protein itself, and also the biological background, microscale thermophoresis MST was used as primary screening tool. So the data are shown here. Assay development and full dose screening took us 10 days. Not shown is the hit validation phase, which takes roughly 5 to 10 days in addition, and also the MedChem SAR support round, 5 days, 5 to 7 per round. So everything starts with a proper QC, not shown here for the sake of time. The protein was qualified. Also, for the sake of time, I cannot show the exact details on the establishment of the controls, but you can see the outcome. So the control was clearly indicating that the protein survives the screening process, and the data are reliable. But the overlap of 16 measurements on 5 days indicate that the noise is rather low compared to the signal. And that's the most important point. This means we can assume that the data were not biased by any data drift or something else. Here's some statistics. Out of the 470, I think, molecules, around about 100 were binding. I have to comment here the rather large hit rate is due to the fact that the fragments were in silico pre-screened. Common hit rates are between 5 and 10%, I would say. And due to the fact that we use a full dose screening in a fixed format, there were 37 molecules with defined KD, basically fitting into the concentration ranges, but a larger fraction also with weaker interaction, and some which might even be more potent. Here's a last overview of the top 10 binders, single-digit micromolar affinities to double-digit micromolar affinities. These molecules are then validated, And then basically these validated hits go into the fragment growth phase. And crystals first, as experts in X-ray, would take over here. And this is also why I would like to hand over the control to Sergey.
All right, thanks a lot, Thomas, for the introduction for the primary screen. My camera is flickering. Is it the same on your side?
Yes, it's cutting in and out a bit.
Okay, so I will turn it off so it's not distracting. All right. My name is Sergey. I'm the founder of the company Crystals First, which is specialized in crystallographic fragment screen, but also high performance crystallography to determine the structures as fast as possible, so structural biology can keep the pace up with medicinal chemistry. And we can cover, of course, structural biology, anything from hit ID to lead opt. We have kind of pioneered the structure first approach, which is in the name of the company, Crystals First. We published recently a paper highlighting this approach, which was fortunately also covered by Derek Loeb in the pipeline. So as a reminder, there is a clear correlation between the release of structural biology on the PDB and number of drugs that have been developed. But how do we get to the structural data? Assuming we have a fragment and would like to determine co-structures, and there are two ways, and the word co-structure kind of implies that there's only one way to do that. It's co-crystallization, where the protein is incubated with a ligand and then crystallized. But there's also another approach called soaking, where the protein is crystallized in an aqueous state and then the crystals are soaked with ligand solutions. And this is where we are specialized in. The data collection and everything after that works very similar. But soaking, in my opinion, is still underutilized way to determine co-structures. I would like to talk about the typical screening cascade and the prioritization of fragment hits, which is kind of driven by biophysical methods, a primary screen, and then structural biology kicks in, and then the optimization cycle begins. And what is critical for this process generally, because like the location of the binding event of fragments is important. It's important to have structural data that guides medicinal chemistry how to grow fragments and apply different fragment growth strategies. But I would like also to kind of challenge the opinion that KD should be the only driver to prioritize compounds for evolution. There is some data published here by Chimera where they had a fragment hit with millimolar affinity, and they could optimize it driven by structural data into highly potent compound, and not only just a binder but a degrader for proteins. And it's kind of highlighting the trend I've been observing in targeted protein degradation. The structural data is driving the design of bifunctional molecules. And we shouldn't be afraid to start with low affinity, kind of very low affinity binders. Same here for SHIP2 by Aztec, starting with 1 millimolar, optimizing to early lead, low micro. And one of the best examples is this publication from Beringer from 2020, where first of all, they kind of we started the company in 2018. I'm happy that they published this paper 2 years later. So they write X-ray first, which is the same as crystals first. But they go a step further and say, after synthesis of the compound, you should determine the co-structure and then the affinity. So it's an interesting example that for 3 compounds that bound with a low affinity, they identify a privileged scaffold and could optimize it quickly, guided by structure to nanomolar compounds. So once again, we shouldn't be afraid to determine co-structures of low affinity binding. There are many more examples of that. So why is X-ray crystallography not used for soaking like X-ray first? The problem is that, as we all know, protein crystallography is very fragile. The protein crystals are very sensitive. They don't like change of the environment. The industry standard to optimize it is trial and error. There are solubility issues with compounds. Fortunately, there is a smart soak technology that we offer where we stabilize protein crystals, increase the quality, the behavior of crystals predictable, and we can work with high solvent concentrations using DMSO, but also other solvents. And as we know, some proteins just don't like DMSO, and we're flexible there. There is a phenomenon called empty structures, and like empty structures are useless for medicinal chemistry in general. I would like to show what is important, like, to avoid it. First of all, what we do before we discuss any project is what I call the soakability analysis, where we quantify the packing, whether the packing of the crystal system we want to use to determine co-structures by soaking is compatible with the ligand space of interest and also the mode of action. So we align the projects with that. And other reason can be that here we have 3 fragments, and when we use cocktail soaking and they bind to the same site, for crystallography this is noise, and then it's difficult to like assign which density belongs to which fragment. And compared to separate soaking experiments, we clearly see the electron density of individual compounds. Another reason could be for empty structures is the soaking concentration, because to get high quality data, we need to oversaturate the system, and low concentrations of low affinity binders like fragments is sometimes, or I've seen many cases actually not enough. So we use as a standard setup high soaking concentration to get a reliable data. And two examples I would like to discuss is so we can apply the technology for non-covalent mode of action. Here we screen a set of natural product like compounds on a kinase. The interesting data is that we had a high hit rate and we had a high sp3 fraction of binders in the ATP pocket. So there are clusters in the ATP pocket of this kinase, and there are clusters on the surface which might represent allosteric or regulatory sites. Here's the distribution of the hits actually of the library, and we see that the sp3 fraction has a different distribution compared to typical hinge binders. We also apply, and it turned out that optimization that we make by using smart soak plays out very well for covalent binders. One example is this human protein where no covalent binders are known. There are only non-covalent binders. We did this experiment. There's a cysteine that is non-catalytic in the active site. We use conventional soaking and a smart soak setup that is independent from the crystallization protocol, and we identify a binder. I am not showing the data, but actually this binder is inhibiting the protein in a functional assay. The readout of such of the data, just to remind, is I would encourage all primary hits to soak and determine the co-structures, because you never know what you see. We've observed formation of pockets, kind of locking conformational states of loops that structural data reveals in other assays don't. So put in as many primary hits as possible into this experiment. To wrap it up, smart soak in a nutshell, we believe we solve the soaking problem in X-ray crystallography. We have an up to 10x accelerated process to get structural data, and we work with over 30 targets. We overcome solubility issues of fragments. We can apply different solvents. The hit rate in a crystallographic screen is high, up to 30%, depends on the nature of the protein, and the success rate of co-structure determination of primary hits or other binders is even higher. And on the right-hand side, just a snapshot of targets we have worked with. And I would like to pass to our next speaker. It's Jan again. Thanks for your attention.
Thanks, Sergey, for this presentation of the method you can use at Crystal first for identification by soaking. Just one slide to summarize where we are in the process from the heat validation, heat identification, heat validation, the fragment growth, and the lead optimization stage. You can find on the process different position for each company. When you have a fragment or a set of fragments identified and characterized by orthogonal validation, different methodologies can be used to optimize the fragment to a lead compound, then to a preclinical candidate. Fragment linking, when you have different fragments in the binding site, and the strategy is to link together your linker for a synergistic approach and to gain in binding energy. Fragment merging, when you have identified different fragments in the binding site with an overlay. You can combine both to increase the potency. And finally, the fragment growing, which is the most straightforward strategy by adding chemical groups to the initial fragment to increase potency with acceptable physical property. And the idea is to maintain the ligand efficiency in your lead optimization stage. Important to know that the fragment linking is relatively difficult, and the structural information are important to help the medicinal chemist. For the fragment merging, could be a good option to find a lead compound. Sometimes the combination of both fragments can be challenging, and the fragment growing is the most common option. Extension of the initial fragments using structural information by addition of groups binding subpockets present in the candidate of interest improve the potency and the selectivity. Just now, an example of the boron chemistry with fragments. Development of boron-containing drugs increased over the last decade, and the approval of the 4 new medicines containing boron in less than 4 years have been generated much interest for many pharmaceutical and biotech companies. At Oncodesign, we have a benzoxaborol library, 272 compounds available for screening. And all these potential hits have been designed in accordance with lead-like properties, including molecular weight, low lipophilicity, H-bond donor, high sp3 fraction, or TPSA. We have at Oncodesign capabilities corresponding to the high standard of pharmaceutical industry, with an experienced team of experts in organic chemistry and medicinal chemists with a strong track record. All new methodologies in organic chemistry can be applied to support the progress of your drug discovery projects. And just to come back to the library of the benzoxaborol available at Oncodesign, we have different series available with a huge diversity on the boron ring or the phenyl ring. And we have an easy access to a large diversity using this scaffold. And benzoxaborol are well known as acting as reversible inhibitors targeting serine in the binding site or tyrosine. And the use of fragment as covalent inhibitors is very interesting field. And in a couple of minutes, Dr. Neil London will present some example of use of fragments acting as irreversible inhibitors. So it's my pleasure to pass the control to Dr. Neil London, and he will show you a very interesting talk about the use of fragment as irreversible inhibitors.
Yeah. Thanks, everyone. I'm happy to be here and present our contribution to fragment-based drug discovery. So our lab, as Jan mentioned, is very much focused on covalent ligand discovery. There are several reasons where covalent ligands are superior or have some advantages over traditional reversible binders. These include better potency, typically due to the formation of the covalent bond, longer duration of action if it's irreversible or reversible covalent, which mean you can dose less, and they can be very selective if your target nucleophile, typically a cysteine residue, is only found in your enzyme of interest and not in off targets. For this reason, several rationally designed covalent successful drugs were approved in the past decade or so, and these includes very challenging targets such as KRAS, as I'm sure most of you know. But discovering these types of covalent ligands is still challenging, and that's where our lab is trying to make a difference. We're developing technologies for covalent ligand discovery. In my postdoc with Brian Shoik at UCSF, we developed a virtual covalent screening platform called DocCovalent. The focus of today's talk will be an empirical covalent fragment screening platform. But we also have a computational protocol to design irreversible inhibitors starting from crystal structures of reversible binders next to a target cysteine, or to design electrophilic covalent peptide binders. And then recently we moved from just designing the binding or just discovering covalent binders into functionalizing these compounds. And a couple of technologies include, for instance, covalent PROTACs. Using reversible covalent chemistry, we're able to both degrade the protein and maintain the catalytic efficiency of PROTACs. And more recently, we described covalent ligand-directed release chemistry, where you can deliver a specific cargo that is released upon covalent bond formation, or in the reverse direction to tag the protein while evacuating the binding site and keeping your protein active, which is very useful for proximity-inducing applications. But today we'll focus on covalent fragment screening. We wanted to explore, my previous speakers presented really nicely why fragment screening is such an important paradigm in drug discovery, but in the context of covalent binding, we thought that doing fragment screen with an electrophile could solve two of the main challenges with fragment-based drug discovery. The first is that the readout is really simple. When you have an irreversible binder, you can use mass spectrometry. And the second is because of the increased potency, you can screen at rather low concentration. So your initial primary hits are already with decent potency. To explore this hypothesis, we manually curated a library of about 1,000 electrophilic fragments from enamine, all commercially available. Our design considerations were to restrict their molecular size, and the average molecular weight of the fragments in the library was 220 Daltons. They all have mild reactivity, with warheads including chloroacetamide, about 75% of the library, and acrylamides, about 25% of the library. And where available, we included libraries of analogs so we can get structure-activity relationship directly from the primary screen. Then our screening process is we incubate the fragments in cocktails of 5 compounds per well at 200 micromolar typically. Then we incubate with a target protein that has cysteine residue for 24 hours, 4 degrees, to allow working with not so stable proteins. And then we read out using intact protein mass spectrometry. And this is how a typical result may look like. In blue, you can see the mass, the deconvoluted mass of the protein where it's not incubated with any compound. And then in green is the spectra with after incubation with a cocktail of 5 compounds. And you can see from this, first by the delta mass between the two graphs, you can see that this was the compound that was adducted to the protein. And also you can assess the percent labeling because we don't see any non-labeled protein left. So this labeled to 100%. And so we can assign percent labeling for each one of the 1,000 compounds in a very short time frame. And we did this in this work for 10 different proteins. In each column, you have a different protein. In each row, you have a different electrophile fragment. And you can see, for instance, if you look at the protein, 6 out of the 10 showed nice hit rates, whereas BSA that we used as a negative control showed barely any hit rates. And these 3 proteins were not amenable to this approach. So the first takeaway message from this is that this is a really easy and rapid method to define covalent tractability of a target protein. So we know that these are very challenging proteins to go after with covalent ligands. But the second, I wouldn't say a surprise, but maybe unintuitive finding was that relatively few of our covalent fragments were very promiscuous. Only about 30 compounds showed more than 50% labeling for 2 or more proteins, or 30% for 3 or more. And if you use a more stringent criteria, only 60 compounds showed more than 30% labeling for 2 or more proteins. And even more importantly, now after we have this data, and by now we ran more than 40 screens with different proteins with this library, we have a database of which are the promiscuous compounds, and we know to ignore them as hits and only follow fragments that are very specific to one protein over other proteins we screened in the past. So this sort of gives us a roadmap on which compounds we should follow up on. One quick example of a project where we utilize this library, in the beginning of 2020, the COVID pandemic just started in China, but a colleague of mine from Oxford University told me they have the main protease of the virus expressed and purified, and it's a cysteine protease, so the cysteine and the binding site, and he asked me if we're interested to screen it using our library. We did, and we got a hit rate of 15%, 150 compounds were able to covalently label the catalytic cysteine at very low concentration because it's very nucleophilic. And then we validated about 40 of them, and they were able to determine crystal structures using soaking, like Sergey mentioned, for about 40 covalent fragments and 40 non-covalent fragments. And here you can see how exhaustively these 80 fragments covered the active site of the main protease of SARS-CoV-2. And all of these different ligands give you a lot of opportunities to optimize and basically improve the potency of the fragment. So here you can see some very easy fragment merges. You can see this this moiety turns into this subpocket, and this moiety goes into this subpocket, but the rest of the molecule is basically the same. Indeed, making the both substitution in one molecule improved its binding affinity, but you can see here a lot of different options. And so we used crowdsourcing. We turned to the community to come up with ideas for merges, and this was one of the merges that we followed up on. This is a covalent fragment with a chlorophenyl. You can see here the covalent bond to the target cysteine. This is a reversible fragment that makes a very important hydrogen bond through this aminopyridine moiety, and this merge basically gave us this initial compound, which is only 25 micromolar IC50 inhibiting the protease, but you can see through a careful medicinal chemistry optimization campaign, we were able to reach to a 40 nanomolar MPRO inhibitor. A close analog of this compound is now under preclinical optimization for in vivo PK and toxicity, and hopefully we'll be able to file an IND sometime next year. So this is a really, I would say, impactful example how even though we ended up not going for a covalent inhibitor from various medicinal chemistry considerations, you can see that the chlorophenyl from the original covalent fragment screen is actually found in the preclinical candidate. But the focus of today's talk is another protein. It's PIN1. It's a unique phosphodependent proline isomerase. It only recognizes phosphoserine or phosphothreonine following a proline residue and isomerizes that proline from cis to trans or vice versa. It has a cysteine in its active site. You can see here cysteine 113, and it controls the stability by this isomerization of various tumor suppressors and oncogenes. The most known one is c-MYC, perhaps NF-kappa B, and Notch, but it really is a signaling hub for controlling many activities of cancer-related proteins. It's found overexpressed in many, many different cancer types, and there are a lot of genetic models where knocking out PIN1 abrogates cancer formation, including on the background of KRAS mutations, p53 mutations, or constitutive MYC expression. However, it's a challenging target since it is a proline isomerase. It was evolved to bind peptides, so you can see it has a very shallow binding site. And even worse, it was evolved to bind phosphopeptides, so the binding site is really positively charged. And all of the previously known binders were negatively charged, which means they had very poor permeability. And in fact, there was no good selective in vivo active probe for PIN1 when we started this project. We targeted this cysteine with a covalent fragment screen, and again, we got a pretty high hit rate of 11% from the primary screen. And out of the top hits, you can see that we had this motif of sulfolene or sulfolane chloroacetamide coming up as over-enriched in the top hits. So we naturally focused on the sulfolane. This is another electrophile we wanted to avoid. And based on modeling, we decided to modify this side chain. I'll spare you the optimization campaign, and here are just the top side chain, top labeling compounds, basically. And here you can see that if we compare on the x-axis the intrinsic thiol reactivity of these compounds to their ability to label PIN1, we don't see any correlation. The fact there's no correlation means that they bind, what matters for labeling is their reversible recognition, and they're not dominated by their thiol reactivity. For this reason, it's always best to choose the lowest reactive hit, and we chose PIN13, or as we named it, sulfopin, which has this tert-butyl side chain. In fluorescence polarization competition assay, it has an apparent Ki of about 20 nanomolar, whereas the version without the chloro that is non-covalent is completely dead. Comparing in an enzymatic, coupled enzymatic assay, it has an apparent Ki of 200 nanomolar, and you can see it's about 2 orders of magnitude better than the so-called state-of-the-art inhibitor juglone, which is in fact not very selective. It's also stable in microsome and has decent oral bioavailability with no toxicity in mice up to 40 milligrams per kilogram when injected once daily. We are also able to determine a co-crystal structure with sulfopin at the active site of PIN1. Here you can see the covalent bond formed with cysteine 113, and this explains nicely the SAR. The sulfolane motif binds in the proline binding pocket, forming 2 buried hydrogen bonds here that are pretty favorable enthalpically, and the tert-butyl chain rests on this leucine 122, but also sterically blocks access to the chloroacetamide, which may explain why sulfopin was less reactive than other substituents at this position. To assess cellular engagement, we synthesized this dead thiobiotin probe coming off of the tert-butyl. It has similar potency in the fluorescence polarization assay, so we could use it to label PIN1 and then compete it with our compound, and we saw very nice target engagement in cells. And to assess selectivity, the selectivity of this in cells, we use 2 chemoproteomics platforms. The first one is Site ID, developed by Jared Marto in Dana-Farber, where we use the dead thiobiotin probe in competition with increasing concentration of sulfopin. And then for each identified proteomic target, you can assess whether you see a dose response or not. And out of 160 identified cysteine sites, PIN1 cysteine 113 was the only one that showed a dose response by more than 2 standard deviation from everyone else. And then using a variation of isotope ABPP called RDTOP, we were able now to identify 2,200 different cysteines. We did this in 2 replicates, and the only cysteine identified in both replicates is cis 113 of pin1. So truly unique selectivity for this cysteine over the rest of the cysteinome. We then tried to recapitulate a phenotype of pin1 inhibition, or rather genetic knockout of pin1 in vivo. So in this paper from Nature Immunology, the author showed that genetic knockout of pin1 reduces germinal centers, increases germinal center size because pin1 regulates BCL6, which is a master transcription regulator in B cells. And then we see here that treating mice pre-immunization with sulfopin has the exact same phenotype of significantly increasing the germinal center size. So this gave us a PD indicator that our compounds are not only orally bioavailable and non-toxic, they also show the desired effect in vivo and phenocopy the genetic knockout of pin1. But we were interested more in the oncology direction. For that, we made knockout cell lines of pancreatic cancer cell line, PA2. Here you can see the growth effect of sulfopin at 1 micromolar in regular cells. You can see there's a significant growth slowing. The compound actually does not kill the cells. We showed that it slows down the cell cycle. So after 8 days, you see a really significant difference in growth. Where if you knock out pin1, the cells grow slower, but now adding sulfopin has no significant effect. So this is an on-target effect, and we have the exact same phenotype in spheroids. So in 3D culture, rather in 2D culture, sulfopin has this effect. The non-covalent control shows nothing, and in the knockout cells, we see no effect. To understand the mechanism more deeply, we did RNA sequencing, which identified about 200 significantly downregulated genes upon treatment. Enrichment analysis showed that MYC signature is being downregulated, which makes sense because it was previously known that pin1 regulates mixed transcription. To show that directly, we used a reporter cell line where we have luciferase under MYC control. You can see here that co-transfecting the cells with pin1 shows a 13-fold increase in the reporter luminescence, whereas after incubation with pin1, we can rescue about half of that transcriptional effect. This led us to test it in MYC-driven in vivo cancer models. One of them was in zebrafish, where MYC is constitutively expressed with GFP in the fish neurons, emulating genetically a neuroblastoma. So here you can see how the GFP looks like. Here you can see how a tumor looks like in this fish. And then upon treatment, just adding sulfopin to the swimming water at these concentration, we can abrogate tumor formation. And this is in a dose-dependent manner. And perhaps in a more challenging experiment, if you take one of these tumors and you transplant them into naive fish, we can also reduce the tumor size upon treatment with sulfopin in the swimming water. In a similar model, genetic model in mice, again, MYC is genetically overexpressed in neurons in these mice. You can see that treatment with sulfopin twice daily, 40 milligrams, can shrink tumors as assessed by MRI after 7 days. You can see this is statistically significant, but even more important, it really extends the overall survival of the treatment group. Again, this is in dose response. The single daily group shows about doubling of the overall survival, and the double dose group showed an even more significant overall survival extension. And finally, in a pancreatic PDX model, we're also able to show tumor regression by treating 40 milligrams per kilogram, and again, extension of overall survival in this very aggressive cancer model, and perhaps most satisfyingly, and this is work that we didn't contribute to in any way other than providing the compound. They showed that if you combine sulfopin treatment with immuno-oncology checkpoint inhibitor and chemotherapy, you can actually cure PDAC in this model. So you can see here that 3 out of 8 mice treated up to 250 days after the implantation were alive more than 100 days after stopping the treatment. So this is really exciting result showing that PIN1 inhibition combination with checkpoint inhibitors can have a real impact on pancreatic cancer. To conclude, I hope I convinced you that covalent fragment screening is a fast way to evaluate the ligandability of a target cysteine to find quality lead against challenging targets, that covalent reactivity is relatively mild and can be dealt with by either by estimating the reactivity of the fragments hits that you have, or by knowing their promiscuity after we screened many different proteins. These fragments can be rapidly optimized, and you're starting from a more potent starting point, and they can be turned into bona fide probes and even preclinical leads. And that covalent binding allows targeting challenging targets such as PIN1 and the main protease. And with that, I'd like to thank the many people that were involved in this project, people from my lab, but also this was a very close collaboration with Nathaniel Gray back at the time at Dana-Farber, now at Stanford University, and Kunping Liu from Dana-Farber, and many other collaborators from Weizmann and other institutions. And I'd like to thank you for your attention, and I'll give the controls back to Sonia.
Okay, well, thank you very much, Nur, for that very insightful presentation. In fact, I'd like to thank Nur, Thomas, Sergey, and Jan for that. And I'm going to now launch our 2 poll questions. I'll run it very quickly, and I'll launch it right now, and you should see it appear on your screen. And there it is. So I'll give you 40 seconds to complete this simple poll question. This is done in real time, so your participation is strongly encouraged and very much appreciated. And once the polls are done, we'll get right into the Q&A. So the question is, have you ever used FBDD in your R&D? Yes or no? So please go ahead and cast your vote. It looks like we got the majority of our audience have cast their votes, so I'm now going to close the polls, and let's take a look at the results. Okay, so here are the results from our audience polling, and we have in our audience 67% said yes, while 33% said no. So thank you everyone for participating in poll question number 1. Now we're going to jump into poll question number 2. And just like before, it's done in real time, so your participation is strongly encouraged again. And here is the question: what is your preferred screening method for fragments? Is it no preference, thermal shift, SPR, NMR, or is it X-ray crystallography? So please go ahead and cast your votes again. And once again, I'll give you about 30 more seconds to complete this simple poll question, and then we'll take a look at the results and get right into the Q&A. Okay, perfect. Thank you so much, everyone. I'm now going to close the polls. Okay, so here are the results. We have in our audience 9% chose no preference, while 17% of our audience said thermal shift. Another 23% said SPR, while 15% said MNR, and then 36% of our audience said X-ray crystallography. So thank you everyone for participating in our 2 poll questions. And now with that, we are going to jump into the Q&A portion of the webinar with the time that we have left. And so I'll invite our speakers to put on their webcams. And here they all come. Perfect, there they are. Hello, everyone. Okay, so while you're speaking, I did receive a bunch of questions that came in. So I'm going to start with this first one here, and I think this question is for Nur. So let me just find it here again where that audience sent it. Okay, here it is. Okay, so the question is there a rule of thumb from your experience, how many covalent fragments from the primary screen by MS covert to co-crystal structures?
I would say the majority. So if we screen for, if we filter for reactivity, so we don't soak high reactive fragments, we get structures for almost all fragments, since we're able to label the protein to 100%. A cautionary warning: if you do soak a reactive fragment, you typically don't get density because it will just flail around after labeling that cysteine. So that's actually, you know, it's another way to evaluate whether your covalent fragment was driven by recognition or by reactivity. So I highly recommend to follow up with the crystallographic soaking.
Sergey, I see you're shaking your head. Do you have anything to add to that before I go to the next question?
Yeah, we've seen. I mean, it's the beauty of academic research. You can talk about the data freely, but what we've seen is exactly what you described. That you see the labeling, but you don't see the recognition. So this explains a lot. Thanks for sharing your experience here.
Okay, perfect. I'm going to squeeze in one more question, if that's okay with everyone. This question here is, what about other amino acids? Can we go beyond cysteine?
Yeah, so cysteine is the most popular, I would say, because it's the most nucleophilic. But recently, there's a lot of interest in developing new chemistries for other amino acids. Lysine, in particular, is seeing a lot of attention with chemistries such as salicylic aldehydes or boronic acids, sulfonyl fluorides. And other chemistries. And now I'm seeing companies slowly are starting to offer fragment or non-fragment covalent libraries for additional chemistry. So for serine, there are a few boronic acids, as Jan mentioned, you can go after with boronates. And carbamates, phosphonates, and these are available through some chemical vendors.
Okay, thank you so much, Nur. You know what? I'm going to squeeze one more in. Okay, sorry about that. This looks like it might be for Thomas. What is your preferred biophysical method for screening of fragments?
Yeah, good question. I think it depends on the project, and I mean, many different tools are available. All have pros and cons. I don't think that I have a real answer on that. So it really depends on the question that you ask. In our portfolio, we have succeeded with all methods, but we also saw problems with all methods. So I think it depends really on what the project will be and how it looks like.
Okay, all right. Let me see. I think we have time to squeeze in another one here. Let me see here. Give me 1 second, everyone. Sorry about that. Okay, I don't see. Oh, sorry. Sergey, maybe this one here is for Sergey again. This one is, you have shown several examples that low affinity fragments can be optimized to highly affinity binders. Do you see a general trend or a shift in the industry towards a structured first approach?
Yeah, I think like what Neil Lodden has presented, also what we see that the biology for drug discovery is becoming more complex and not trivial. We have challenging targets, so-called undruggables, and I think we see a shift that structural data is driving drug discovery, especially what I mentioned, if you look at PROTACs, where the geometry and the conformation of ligands plays a crucial role for optimization cycles. So yeah, I see a trend, and I think with DeepMind's release of their predicted structures, there will be more experiments that come from AI-driven companies that have to be validated also in an experimental setup.
Okay, thank you so much, Sergey. Well, that's all the time we have. We have so many questions that have come in from our audience members, but unfortunately our time has run out. So I just wanted to let you know, if we could not attend to your questions, the team at OncoDesign will follow up with you after this presentation. And if you have any further questions, please utilize the email addresses that are on your screen, and I'm sure they'll be happy to reach out to you. So thank you everyone for participating in today's webinar. You will be receiving a follow-up email from X-Talks with access to the recorded archive for this event. A survey window will be popping up on your screen. Your participation is appreciated as it will help us to improve on our further webinars. Now, give me a few seconds. I'm going to send you a link in your chat box, and you'll be able to view the recording of this event on this page. And you can also share this link with your colleagues once they register for the recording as well. So I encourage you to do that. Now, please join us in thanking our speakers, Thomas, Sergey, Nur, and Jan, for that very insightful presentation. And thank you so much for answering our audience questions. I'm sure they appreciate that. Thank you, gentlemen. It has been my pleasure to be your webinar moderator. On behalf of the team here at X-Talks, we thank you for joining us. I'm Sonia Hunt. Until next time, please take care and bye for now. Bye, everyone. Thank you.
Bye.
Bye-bye.